Today we are going to discuss the physiology of hemostatic process. We all know that blood remains in the fluid state in the blood vessel lumen. But whenever there is an injury to the blood vessel, the subendothelial tissue gets exposed and there is a formation of the blood clot. Cessation of blood loss from a damaged blood vessel is known as hemostasis, which involves primary hemostasis and secondary hemostasis. Primary hemostasis is the initial phase of hemostasis. Here, there is the formation of platelet plug, that is, white plug is formed at the site of injury. Here, the platelet adheres, they become activated and aggregate to form a temporary seal. Followed by primary hemostasis, we have secondary hemostasis, which involves the activation of coagulation cascade, which ultimately converts fibrinogen to fibrin. This fibrin is responsible for clot stabilization, which seals the vessel more permanently. So let's see the platelet aggregation. Platelets have two types of receptors, that is group 6 and group 1b. Whenever there is an injury to the endothelium, the subendothelial surface gets exposed. So there is exposure of collagen and von Willebrand factor. Group 6 receptor present on the platelet, they bind to this collagen, and group 1b binds to the von Willebrand factor, and the platelets adhere to the subendothelium surface of the damaged blood vessels, which activate the platelet, and there is a release of aggregates, that is thromboxane A2, ADP and serotonin. This released ADP then stimulates P2Y1 and P2Y12 receptors on the other platelets and activates this glycoprotein 2B3A receptors and increases the COX-1 formation, which further promotes the platelet aggregation, and there is a binding of these aggregates to fibrinogen. Exposed phospholipid from the platelet surface promotes thrombin formation in the coagulation pathway. This thrombin then acts on protease activated receptor type 1 and type 4, which promote platelet aggregation. Thromboxane A2 is the major product of COX-1, which is involved in the platelet aggregation, and prostaglandin I2, that is prostacyclin, which is synthesized by the endothelial cells. This prostacyclin, they inhibit platelet aggregation. The balance between thromboxane A2, which is released from the platelet, and prostacyclins, which is released from the vessel wall, is necessary to control the intravascular thrombus formation. Platelet aggregates, they form the white plug, which is soft and labile and which is unstable at the site of injury. To make it more stable, there is a stimulation of coagulation cascade, which increases the release of clotting factors, which converts this fibrinogen, which is soluble or unstable, to fibrin, which is insoluble, and stabilize the clot. So further we'll come across the coagulation pathway. So at the site of injury, there is a release of tissue factor along with factor 7A and calcium. It activates factor 10. This activated factor 10 along with factor 5A and calcium converts prothrombin to thrombin. This thrombin then converts fibrinogen to fibrin. This fibrin, which stabilizes the clot. So naturally there is a presence of anticoagulant pathway, that is antithrombin 3, which breaks down or inactivate this clotting factor such as 2A, 9A, 10A, 11A and 12A. So for this fibrin which is formed, they are first secreted as monofilament. After stabilization, the fibrin gets intertangled through glycoprotein 2b 3a of platelets, and this platelets gets trapped in the meshwork and forms a clot. To prevent the excess clot formation, the fibrinolytic pathway gets stimulated. So at the site of injury, the endothelial cells also secrete the tissue plasminogen activator. This tissue plasminogen activator binds to plasmin only in the presence of fibrin, which converts plasminogen to plasmin. This plasmin digests the fibrin or breaks down this fibrin. Also, the plasmin activator inhibitor type 1 and type 2, they inactivate this tissue plasminogen activator, and alpha 2 antiplasmin, that is alpha 2 AP, they inactivate this plasmin. So ultimately the platelet aggregation, they form the white plug which is soluble clot, which forms a temporary plug. To make it permanent, coagulation pathway gets activated, where there is a formation of red plug or insoluble clot is formed. To prevent this excess clot formation, the fibrinolytic pathway gets stimulated, and the balance between this coagulation and fibrinolysis is necessary to prevent thrombosis and bleeding, because both, if present in excess, can be fatal. So in arteries, the platelet mass is the main constituent of the thrombus, and here there is a formation of white plug. So antiplatelet drugs are effective in arterial thrombosis, while in veins, due to sluggish blood flow, a fibrinous tail is formed which traps the RBC, and thus a formation of red plug or fibrin tail is formed. So in case of venous thrombosis, anticoagulants are more effective. Thank you.